Nanaimo, British Columbia, Canada, Mission 12, semi-finalist proposal.
Hello, bonjour. I'm Erin Burnley.
I'm Katja Hannison. I'm Melanie Deifran. I'm Maya Dunn. I'm Leslie Ferrier.
And we are from Nanaimo, British Columbia, Canada. Our experiment was designed to answer the question of whether microgravity influences the virulence of Cucuria rhizophila bacteria using Caenorhabditis elegans. The C. elegans model of infection was chosen because it is an established model for studying infection in eukaryotic systems, as they are anatomically simple and genetically similar to humans. Cucuria rhizophila is a level 1 bacteria that is present on 96% of all humans. This bacteria poses a particular risk to astronauts, as it is an opportunistic pathogen for those with compromised immune systems and can lead to more serious infections, such as meningitis and pneumonia. Additionally, NASA has identified Cucuria rhizophila as a problem bacteria on which minimal research has been conducted relating to microgravity. Space microbiology is a relatively new field, and the insight gained from this experiment can help create safer conditions for space exploration, adjust healthcare for astronauts, and add to the growing body of knowledge on how microgravity affects all forms of life. Researchers have conducted similar experiments using different bacteria and testing their virulence fluctuations. These studies came back with varying results, wherein some bacteria became more virulent and some bacteria became less virulent.
Regarding Cucuria rhizophila, previous experiments have suggested that its growth is more rapid in simulated microgravity, but its virulence has yet to be tested. Based on this research, we believe that the virulence of Cucuria rhizophila will be altered whilst in microgravity.
To begin our experiment, we put the C. elegans into a dour state. In this state, the C. elegans are developmentally arrested and can survive in the absence of food for months. Furthermore, C. elegans can be induced to exit dour and resume normal growth by exposure to a suitable food source, which will be done once they're in space. By this method, effects of the spaceflight to the ISS can be eliminated. We used the wet plate protocol to induce dour state. A centrifuge was used to spin the worms into a pellet, and the supernatant was removed. We then added 20 milliliters of fresh E. coli grown in Luria broth medium into a petri dish. Then we added 2 milliliters of the C. elegans and the Luria broth mixture onto a freshly poured 10 centimeter nematode growth medium plate. The plates were then left in an incubator for 10 days at 25 degrees Celsius to allow worms to enter dour state. Once the worms have been induced into dour, we will add 0.5 grams of the nematode growth medium, including the C. elegans, into the test tube, and a dissolvable hypermellus capsule containing 0.07 milliliters of Cucuria rhizophila bacteria. In the second compartment, we will add 2.8 milliliters of the tryptic soy broth, and finally, in the third compartment, we will add 2.8 milliliters of the formalin. 5 days before undock, the astronaut will remove the first clamp and shake the tube for 60 seconds. The broth will dissolve the capsule, releasing the bacteria, and the C. elegans will consume the bacteria, awakening them from their dour state. 2 days before undock, the second clamp will be removed, and the formalin will kill the remaining C. elegans. Once the test tube is returned to Earth, we will analyze the data under a microscope using accounting software to count the amount of needle C. elegans and curled C. elegans.
The following procedure describes the steps used in our clinostat trial and ground truth. We first induced dour in the C. elegans using the wet plate protocol. We then coated the bottom of the test tubes with 2 milliliters of nematode growth medium and 0.5 milliliters of dour C. elegans in the Luria broth. After 2 days, we added 0.5 milliliters of tryptic soy broth and Cucuria rhizophila. We used 2 test tubes as ground controls. After 3 days, we added 2 milliliters of formalin, terminating the experiment. Simultaneously, the 2 experiment tubes were attached to the 3D clinostat to simulate microgravity. After 3 days, the 3D clinostat was paused, and we removed the test tubes. We then added 2 milliliters of formalin, terminating the experiment. A 3D clinostat, or random positioning machine, is used in screening or testing to simulate a microgravity environment for small organisms. It uses 2 gimbal-mounted frames attached to separate motors. The outer frame rotates at a constant speed of 6 revolutions per minute, while the inner frame rotates randomly between 5.5 and 6.5 revolutions per minute. This causes the sample to feel as though 1G points in all directions, as they have no down reference point. This can be confirmed by the assumption that gravity must point in a certain direction for a minimum amount of time in order for small organisms to register it.
As previously stated, we expected the virulence of the Cucuria rhizophila bacteria to be altered in microgravity. Unfortunately, because all of the Caenorhabditis elegans, both in the ground experiment and in the simulated microgravity experiment, were found dead, our results were inconclusive. In both the ground and microgravity experiments, it was clear that the C. elegans had been killed by the bacteria because they were all needle-shaped. Had they been curled, we would have known that they had been killed by the formalin, because the formalin kills the worms faster than the bacteria, so they are still moving around when they die. As displayed in 2 of the photos, a C. elegans is needle-shaped. killed by the bacteria in the ground experiment. The third photo displays a C. elegans in its natural movement. This particular organism was not used in the ground experiment or in the simulated microgravity experiment.
The results from our experiment showed that all the C. elegans were killed by the bacteria Cucuria rhizophila. Since the bacteria was so virulent on Earth, we were unable to tell whether the bacteria was in fact more or less virulent in a microgravity environment, so we were unable to prove or disprove our hypothesis. In future research, we would explore methods to either decrease the virulence of the bacteria or increase the hardiness of the C. elegans. If we were to do this experiment on Earth again, we would terminate the experiment in 1 day instead of 3 to ensure that some C. elegans would still be alive and we could actually compare our microgravity and ground experiments. Thank you everyone for listening. Merci d'avoir écouté. Are there any questions?
So my question is that you guys said that there was previous research on virulence factors, and some, got more intense, some got less intense. Are there certain virulence factors that are like, if it's an enzyme or if it's this or if it's breaking down the blood, that seem to be more prominent in the zero gravity than Earth? Can you tell based on whatever that factor is? Is it kind of consistent, I guess, overall?
There has been research that have been done on several types of bacteria, including Candida albicans, Staphylococcus aureus, and Listeria monogenous. And unfortunately, at this point, there are no connections in the research. Some became more virulent, some became less virulent, but the question is still why. And unfortunately, based on the science and the research that we based our experiment upon, there was no connections.
Thank you.
Thank you, ladies.